College, University, Faisalabad. Amino acids. Protein forming standard amino acids. Non-protein forming non-standard amino acids. Protein forming amino acids. Essential amino acids. Essentials amino acids. Methylphenidate, methionine, arginine, threonine, valine, isoleucine, leucine, phenylalanine, histidine, and lysine. Non-essential amino acid: alanine, asparagine, aspartic acid, cysteine, glutamic acid, glycine, hydroxylysine, proline, serine, and tyrosine. Non-protein forming are non-standard amino acids, special amino acids. Special amino acids are citrulline, ornithine, tyrine, dopa, GABA, and etc. Special amino acids. These amino acids are produced in our body but do not take part in protein synthesis. They have special functions. Aromatic amino acids. These amino acids contain aromatic or benzene ring in their molecules. Sulfur containing amino acids. They contain sulfur in their structure. They provide sulfur to the body. Examples are methionine and cysteine. They take part in the formation of disulfide bonds. Two molecules of cysteine join together to bisulfide bond and form cysteine. Acidic amino acids. They have more carboxyl group than amino group. They are monoamine dicarboxylic acid. Side chain of these amino acids are proteins or H donors. Basic amino acids. They have more amino group than carboxyl group. They are diamino monocarboxylic acid. Side chain of these amino acids are protein acceptors. Branch chain amino acids. Mnemonic will. Valine, isoleucine, and leucine. Only three amino acids are in this group. They are metabolized primarily in peripheral tissues, muscles, rather than in liver. All of them have a similar routes of catabolism degradation. Amino group. They have amino group instead of amino group in their structure. Glucogenic amino acid, GAG, SHAG, PAC, and MTV. These amino acids are termed glucogenic or glycogenic because upon catabolism they yield products which can synthesize glycogen in liver and muscles through a metabolic pathway called gluconeogenesis, synthesis of glycogen from non-carbohydrate sources. The catabolic products of these amino acids are pyruvate and a product of glycolysis and are one of the intermediate products of citric acid cycle. The pyruvate and the intermediate products of citric acid cycles are the substrate for gluconeogenesis. These include five essential and nine non-essential amino acids. Ketogenic amino acid. These amino acids are termed ketogenic because upon catabolism they produce ketone bodies. Acetostate are one of the precursors such as acetyl CoA or acetoacetyl CoA. Leucine and lysine are the only two essential amino acids which are exclusively ketogenic. Acetoacetate is one of the three ketone bodies. The other two are beta hydroxybutyrate and acetone. Amino acids which have both glucogenic and ketogenic, which are mnemonic T-tip. Degradation of these amino acids yields intermediate products both for glycogen and precursors. These include three essential and one non-essential amino acids. Polar amino acids, mnemonic give calcium stat. These include polar, water soluble, or hydrophilic, or side chain or groups. They include one essential and five non-essential amino acids. These essential amino acids have zero net charge at physiological pH 7.4. At alkaline pH, cysteine and tyrosine can lose a proton. These amino acids participates in the formation of hydrogen bonds. Cysteine, a sulfur-containing amino acid, participates in disulfide bond formation. In aqueous environments such as GIT, the polar side chain R groups are found on the exterior of the protein interacting with the aqueous environment. In lipid environments such as membranes, these polar side chains are clustered in the interior of the proteins. Nonpolar amino acids. Mnemonic Paul give me tryptophan. These contain nonpolar lipid solubles, lipophilic or hydrophobic side chain or groups. They include six essential and three non essential amino acids. Monocarboxylic acid, these are called neutral amino acid. Monoamino dicarboxylic acid, acetic amino acid. Diamino monocarboxylic basic amino acid and amino acids. Monoamine amino monocarboxylic neutral amino acids. These are the amino acids with aliphatic side chains. Examples are glycine, alanine, valine, leucine, isoleucine. Amino acid with aromatic side chains, phenylalanine, tyrosines, and tryptophan. Side chains containing sulfurs, methionine, cysteine. Side chain with OH group, serine, and threonine. Monoamine dicarboxylic acid, amino acid, acetic amino acid, aspartic acid, glutamic acid, lysine, and hydroxylysine. And amino acid, proline and hydroxyproline. Amino acids, uh, there may be ketogenic amino acid, glucogenic amino acid, ketogenic and glucogenic amino acids, polar amino acids, and non-polar amino acids. Glucogenic amino acid, glutamate, alanine, glutamine, serine, histidine, asparagine, glycine, proline, aspartate, cysteine, arginine, threonine, valine. Ketogenic amino acid, leucine and lysine. Glucogenic and ketogenic amino acid, tyrosine, tryptophan, isoleucine, phenylalanine. Polar amino acids, glutamines, glycine, serine, threonine, asparagines, and tyrosine. Non-polar amino acids, proline, alanine, leucine, glycine, isoleucine, valine, methionine, tryptophan, phenylalanine. R group side chain of these amino acids do not bind or give off protons and do not participate in hydrogens or ionic bonds formation. Since the R group of these amino acids are hydrophobic, they participate in hydrophobic interactions by forming non-covalent bonds. In aqueous environments such as GIT, the non-polar side chain R groups of amino acids tend to cluster in the interior of the protein. This is due to the hydrophobicity of the nonpolar R groups. In the lipid environment, such as the cell membrane, the nonpolar side chains R groups are found on the surface of the proteins interacting with the lipid environment. Proline differs from other non-polar amino acids in that it contains an amino group rather than an amino group. Zwitterions, dipolar ions. When an amino acid is dissolved in an aqueous environment, one hydrogen atom moves automatically from the carboxyl group of amino acids towards the amino group of the same amino acids, making the amino acid positively as well as negatively charged dipolar at the same time. Such ion is known as Zwitter ion. A Zwitter ion can act as an acidic acid as well as base depending upon the conditions. Such ions are electrically neutral. Alanine is an example of Zwitter ion, which can act either as an acid proton donor or base proton acceptor. Substances having this two-way property are amphoteric and are often called as ampholites. Isoelectric pH. The pH at which an amino acid has no net charge on it and therefore does not move towards the electrodes in an electric field is known as its isoelectric pH. The isoelectric pH is denoted by Pi. The examples of Pi of amino acids are alanine, aspartic acid, arginine. Examples of Pi of proteins are casein 4.6 and lactalbumin 6.4. At pH below pi, the amino acids donates H plus ions, thus acts as an acid. At pH above pi, the amino acid accepts H plus ions, thus acts as a base. Functions of important amino acids which take part in protein synthesis: methionine. The active form is S-adenosyl methionine. This contains sulfur and provides it to the body. It is a lipotropic factor. It is an initiator of protein synthesis. It donates methyl group to form other compounds. It donates its methyl group to noradrenaline to synthesize adrenaline. Cysteine. It contains sulfurs and provides it to the body. Cysteine. It contains sulfur and provides it to the body. It takes part in the synthesis of glutathione, glycine. It conjugates with the bile acid, cholic acid to synthesize bile salts, sodium glycocholate. In liver, it detoxifies benzoic acid by converting it into hippuric acid, which is then excreted in urine. This is the detoxification action of glycine. It takes part in the synthesis of glutathione. It takes part in the synthesis of heme, purine, and creatine. It can be converted to other amino acids, serine, which is converted to pyruvic acid and ultimately to glucose. Glycine, therefore, is glucogenic. It is a major inhibitory neurotransmitter which is used by the uh, Renshaw cells of the spinal cord. Tetanus toxin causes muscle spasm by inhibiting the release of glycine. Aspartic acid. It takes part in the synthesis of purine and pyrimidines basis for nucleic acid. It is a major excitatory neurotransmitter of the chain. Glutamic acid. It takes part in the synthesis of glutathione. It is a major excitatory neurotransmitter of the brain. Glutamic is glutamate via its receptors play a role in long-term potentiation of hippocampal neurons, a memory process. Phenylalanine. It synthesizes tyrosine, dopamine, and adrenaline. Tyrosine. Tyrosine synthesizes thyroid hormones. Thyroxine synthesizes neurotransmitters, for example, noradrenaline, and adrenaline synthesizes melanin. Tryptophan. 60 milligram of tryptophan in the body produces 1 milligram of niacin, vitamin B3. It synthesizes serotonin. Proline. It is a major constituent of connective tissues such as hydroxyproline in globin, in collagen. Amino acids which do not take part in protein synthesis, citrulline and ornithine in urea cycles. Both of these amino acids are used in the formation of urea from ammonia. Tyrine. It conjugates with bile acid, cholic acid to synthesize bile salt, sodium tyrocholate. GABA, it acts as a neurotransmitter. DOPA, it acts as a neurotransmitter. It is used therapeutically in the treatment of Parkinson's disease. The amine form, dopamine, also acts as a prolactin inhibitory factor. Interconversions of amino acids. Many amino acids can be converted to other amino acids. Phenylalanine to tyrosine to dopa, methionine to cysteine to cysteine, cysteine to alanine, glycine to serine. Peptide bond, acid amide linkage. We know that proteins are made up of amino acid. In a protein, two amino acids are joined together by carboxyl group on one side and amino group on the other side through covalent bond, forming an acid amide linkage known as peptide bond. Peptide bond is extremely stable and is only cleaved by proteolytic enzymes. Peptides. Peptide means amino acids. Oligopeptides are the peptide chains made up of 2 to 10 amino acids linked through peptide bonds, disulfide, dipeptides, tri and tetrapeptides. Polypeptides are the peptide chains made up of more than 10 amino acids linked together by peptide bonds, albumin, globulins, globin. Organization of protein structures. The structure of proteins are organized at four different levels: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure. The number and order of amino acid in the straight polypeptide chain is called the primary structure of the proteins. Secondary structure, the folding of the polypeptide chains into a specific coiled structures held together by disulfide and hydrogen bond is called the secondary structure of the protein. tertiary structure, the arrangement and interrelationship of the coiled chains of proteins into specific layers or fibers is called the tertiary structure of the proteins. Quaternary structure, the several monomeric units, each with the appropriate primary, secondary, and tertiary structures, may combine together. The association of similar or dissimilar subunits is called the quaternary structures of the proteins. Sources of proteins: animal sources, meat, fish, egg, and milk. Plant sources, pulses, legumes, and meat. First class proteins. These are the proteins which contain all essential amino acid protein from animal source. And second class proteins, these are the proteins which do not contain all the essential amino acid protein from plant source. How can we get all the essential amino acid from second class proteins? By mixing two different kinds of the second class proteins, we can form first class proteins.